Hello guys, good afternoon. Welcome to the last presentations for General Lab Biology. Today we are gonna present about meiosis and mitosis. Our group member consists of Viola, Indah, Stevani, and me, Feby. Alright, here we go. Here is the table of contents of our presentations. The first one, we are talking about what is the cell divisions and why is it important for us? Second, the definitions and the phase of meiosis. Next, observing meiosis in laboratory. The fourth one is the definitions and the phase of mitosis. The last one is observing mitosis in laboratory. Initially, let's talk about what is cell divisions and why is it important? The definitions of the cell division is the process by which cells multiply involving both nuclear and cytoplasmic division. Compare meiosis and mitosis. Most of the time, when people refer to cell division, they only mean mitosis, which is the process of making new body cell. Meanwhile, for the cell divisions, there is also meiosis, that is, cell division in order to create egg and sperm cell. Then, why cell division is important? First, we are made of cells. Not only we, but all living things are made of cells. Second, cell is basic unit of structure and functions of living things. Next, maintain of genetic diversity. Cell division, especially the meiosis, ensures that all living organisms will maintain genetic diversity. Fourth, we need to keep our cell to stay alive. We are living organisms that made of cells. We need to keep living. Therefore, our cells must stay alive. In order for a cell to keep living, our cell must divide and multiply. Last but not least, all cells come from pre-existing cells. Okay, for the next will be presented by Viola.
Thanks to Feby, now I will explain about the definition and the phase of meiosis. Meiosis is a process where a single cell divides twice to produce 4 cells containing half the original amount of genetic information. These cells are our sex cells, sperm in males, eggs in females. Meiosis contributes to genetic variety. It doesn't make body cells, it makes egg and sperm cells, as well known as gametes. You may recall that humans have 46 chromosomes. That is the number of chromosomes that most people have. Meiosis is what we call reduction division because you have starting cell from 46 chromosomes and your ending cells will be the sperm, and the egg cells has only 23 chromosomes. Meiosis can be divided into 9 stages. These are divided between the first time the cell divides, or meiosis 1, and the second time it divides, or meiosis 2. First, we have interphase. Meiosis starts with the interphase, so the interphase itself is broken up into different phases. The first phase of the interphase is G1 phase, which is the period before DNA gets synthesized. During this phase, the cell grows to prepare for cell division. An important thing to note is that G in G1 stands for gap. And the next part of the interphase is the S, or synthesis phase. In the S phase, the DNA gets synthesized in happens fairly quickly, and the final phase is the G2 or second gap phase, and this is the part where the cell synthesizes protein and continues to grow, and this continues to happen until the start of the next phase. And next we have prophase 1. Immediately after the interphase starts the prophase, there are several events that happen in the prophase. The first event involves the chromosomes condensing and attaching themselves to the nuclear envelope of the cell. Next, synapsis occurs, which is when a pair of chromosomes line up together. When this happens, tetrads are created, which are made of 4 chromatids. During this phase, it is possible for crossing over to happen where the chromatids are moved and possibly rearranged. After crossing over, the chromosome starts to get thicker and break away from the nuclear envelope, and the centrioles will then move away, and both the nucleoli and nuclear envelope will begin breaking down. This process is similar to mitosis. And another one, we have metaphase 1, and the metaphase is pretty simple. The tetrads align at a plate, and the centromeres are all oriented towards opposite poles of the cell. Next we have anaphase 1. The anaphase happens next, and it involves the chromosomes moving to opposite the cell walls. The way this process works is through microtubules that pull the chromosomes and move them around the cell. And we have telophase 1. The telophase continues the processes of the previous phase in that the chromosomes are continuing their movement to the poles of the cell. After the movement is complete, each pole gets a haploid or a complete cell of chromosomes. When the telophase ends, there are 2 daughter cells that are made. Each of these cells has exactly half the number of chromosomes from the original cell. There can be many different processes that happen in this phase in order for the cell to prepare for the second part of meiosis, and the only constant is that the genetic material doesn't replicate, but instead it continues to get divided. And another one, we have prophase 2. The prophase 2 starts again in meiosis, and the process involves the continual breakdown of both the nuclear membrane and the nuclei, and the spindle network also appears. Again, it is important to note that the chromosomes don't replicate anymore, but they instead are simply broken down and separated further. During this phase, the chromosomes start migrating to the metaphase plate, which is located at the midpoint of the cell. Next, we have metaphase 2. The second metaphase is just like the first. The chromosomes simply line up along the center of the cell before it splits into 2. Again, the chromatids point toward the opposite ends of the cell. And the last, we have anaphase 2. The second anaphase is the penultimate phase in meiosis, and it has a few events that occur. The first event involves the sister chromatids separating and moving towards the opposite ends of the pole. The spindle fibers at this time will lengthen and enlarge the cell. The paired chromatids will then begin to separate from each other. Every chromatid pair is considered to be a full chromosome. Just it for me. Next will be continued by Stevani.
Okay, the next one, we will continue to observing meiosis in laboratory. In this video, we will see the experiment from onion flower or pollens of onion. We will see the process of onion flower division in sexually reproducing organism whereby the chromosome within a diploid cell duplicate and separate during the formation of sex cells to finally form 4 haploid daughter cells. and each cell would containing half the number of chromosomes found in the parent cells. Okay, without further ado, let's watch the video. As we already mentioned, there are two types of cell division. The first one is meiosis and the second one is mitosis. And now let's talk about mitosis. So what is mitosis? Mitosis is a process of cell duplication or reproduction, during which one cell gives rise to two genetically identical daughter cells. Strictly applied, the term mitosis is used to describe the duplication and distribution of chromosome, the structure that carry the genetic information. And the major purpose of mitosis is for growth and to replace worn out cells. And the next slide, we will continue with my friend Indah.
So thank you, Stevani, for the time. Let's continue to the seven content of this presentation. It is about the phase of the mitosis. The phase or the stage of mitosis and cell wall is divided by seven phases. The first is interphase, the second is prophase, the third is inlet prophase, the fourth is metaphase, the fifth is anaphase, and the sixth is telophase, and the last one is cytokinesis or daughter cells. So what about the process? Let's we discuss in the next screen. Okay, now let's talk about mitosis process. So the first is interphase. Before a dividing cell enters mitosis, it undergoes a period of growth called interphase. About 90% of a cell's time in the normal cell cycle might be spent in interphase. So there is two phases in interphase itself. The first is G1 phase, that is about the period prior to the synthesis of DNA. In this phase, the cell increases in mass in preparation for cell division. The G1 phase is the first gap phase. The second is S phase. This is about the period during which DNA is synthesized. In most cells, there is a narrow window of time during which DNA is synthesized. The S stands for synthesis. So G2 phase, this is about the period after DNA synthesis has occurred, but prior to the start of prophase. The cell synthesis protein and continues to increase in size. The G1 phase is the second gap phase. So, in the latter part of interphase, the cell still has nucleoli present. So, the nucleus is bounded by a nuclear envelope, and the cell's chromosomes have duplicate but are in the form of chromatin. Next to the second phase or stage in the mitosis, that is prophase. In prophase, the chromatin condenses into discrete chromosomes. The nuclear envelope breaks down and spindles form at opposite folds of the cell. Prophase versus interphase is the first true step of the mitosic process. So during prophase, a number of important changes occur. The first occur is chromatin fibers become coiled into chromosomes, with each chromosome having two chromatids joined at the centromere. Second, the mitosic spindles, composed of microtubules and protein, form in the cytoplasm. Next is the two pairs of centrioles formed from the replication of one pair in interphase. Move away from one another toward opposite end of the cell due to the lightening of the microtubules that form between them. Next is polar fibers, which are microtubules that make up the spindle fibers, reach from its cell pole to the cell's equator. Next, kinetochores, which are specialized regions in the centromeres of chromosomes, attack of a type of microtubules called kinetochore fibers. So the kinetochore fiber interact with the spindle polar fibers, connecting the kinetochores to the polar fibers. So the last is the chromosome begin to migrate toward a cell center. Next to the next phase is about metaphase. In metaphase, the spindle react maturity of the chromosomes lie at the metaphase plate. That's about a plane that is equally distant from the two spindle poles. During this phase, a number of changes occur. First, the nuclear membrane disappears completely. Second, polar fibers, microtubules that make up the spindle fibers, that we talked before, continue to extend from the poles to the center of the cell. The third is chromosomes move randomly until they attack at their kinetochores to polar fibers from both sides of their centromeres. The next is chromosomes align at the metaphase plate at right angles to the spindle poles. And the last change occur in this metaphase is chromosomes are held at the metaphase plate by the equal forces of the polar fibers pushing the centromeres of the chromosomes. Next stage or phase is anaphase. In anaphase, the parent chromosome or the sister chromatids separate and begin moving to opposite ends poles of the cell. Spindle fibers not connected to chromatids lengthen and elongate the cell. At the end of anaphase, each pole contains a complete compilation of chromosomes. During anaphase, it also the important changes occur. The first is the parent centromeres in each distinct chromosome begin to move apart. On the parent sister chromatids separate from one another, it is considered a full chromosome. They are referred to as daughter chromosomes. Next change occur is through the spindle apparatus. The daughter chromosome move to the poles at opposite ends of the cell. And next, the daughter chromosome migrate centromeres and the kinetochore fibers become shorter as the chromosome near a pole. And the last is in preparation to telophase, the next stage of mitosis. The two cell poles also move further apart during the course of anaphase. At the end of anaphase, each pole contains a complete compilation of chromosomes. Move to the next stage is telophase. In telophase, the chromosomes are cornered off into distinct new nuclei in the emerging daughter cells. In the stage, the following changes occur. The first is the polar fibers continue to lengthen. The second is nuclei begin form at opposite folds. Next, the nuclear envelopes of these nuclei from remnant pieces of the parent cell's nuclear envelope and from pieces of the endomembrane system. Next is nucleoli also reappear next to chromatin fibers of chromosomes uncoil. And the last is after this change, telophase or mitosis is largely complete. The genetic contents of a cell have been divided equally into two. So finally we come to the last phase or stage in mitosis, that is cytokinesis. Cytokinesis is the division of the cell cytoplasm. It begins prior of the end of mitosis in anaphase and completes shortly after telophase. The end of cytokinesis, two genetically identical daughter cells are produced. These are diploid cells, with each cell containing a full complement of chromosomes. So that is about the phase and the process of mitokinesis. Okay, for more detail of the mitosis process, we provide you the video of mitosis in onion root tip experience. In this video, it will be preparing root tips of allium species to observe cell in various stages of mitosic division. As known that mitosis is one of the stages in the cell cycle where the chromosomes of a cell replicate and separate to eventually form two genetically identical cells. Unlike the meiosis before, chromosome number is maintained in both daughter cells. For your information, active mitosis is allium species occur twice in 24 hours period, with a primary maximum phase at around 11 p.m. and a secondary maximum phase at around 1 p.m. So based on this information, it goes without saying that these two time periods will be the most preparable for collecting root tip for mitosis lab work. So without saying anything again, let's watch the video.
In this video, we'll be preparing root tips of allium species to observe cells in various stages of mitotic division. Mitosis is one of the stages in the cell cycle where the chromosome of a cell replicate and separate to eventually form two genetically identical cells. Unlike in meiosis, chromosome number is maintained in both the daughter cells. For the experiment, you will need onion or garlic bulb, beakers, toothpicks, carnoise fluid, 70% ethanol, 1 normal hydrochloric acid, acidocarmine or acidoorcine stain, glass slides and cover slips, blade or scalpel, watch glasses or petri dishes, tiny vials or containers, spirit lamp or Bunsen burner or any flame source, blotting paper, droppers, thumb forceps and a pair of scissors, compound light microscope, and immersion oil. To begin the experiment, take an onion or garlic bulb and fix it on a beaker containing tap water using toothpicks like you see here. Make sure the base of the bulb touches the water level. Keep this in a safe corner for a couple of days. Once the roots have grown about 2 to 3 centimeters, cut out 1 centimeter of root tips and transfer them into a tiny vial containing carnoise fixative fluid. Carnoise fluid fixes the DNA of the root tip cells. Leave the roots in the fixative for about 48 hours. If you're planning on storing the root tips for a longer time for use in future, you may transfer the fixed root tips in a second vial containing 70% ethanol. Ethanol dehydrates the root tissue and thereby preserves DNA. Root tips preserved in ethanol and preferably refrigerated will keep well for a couple of years for genetic studies. Either ways, take a few root tips from carnoise fluid or ethanol and transfer them onto a watch glass containing 1 normal hydrochloric acid. Acid will soften the cell walls and weaken cellular connections so it becomes easy to squash the root material later. Gently warm the watch glass on a flame for about 5 seconds. Expose the root tips in the acid for about 2 minutes. Give the root tips a couple of washings in distilled water. Now transfer the roots onto a watch glass containing acidocarmine or acidoorcine stain. These are excellent chromosomal stains and will impart a deep red coloration to the nuclear material of the root cells. Warm the stain on the flame for about 5 seconds and leave the root tips in the stain for about 5 to 10 minutes. Now transfer the root tips onto a clean glass slide containing a drop of water. Using a sharp blade or scalpel, remove about a millimeter of the root tip and discard the rest. The very tips of the roots are the regions with active cell division, and they are all that we require for mitotic experiments. Gently lower a cover slip on the root tips, making sure to avoid any air bubbles. Using the blunt end of a forceps or a pencil, gently tap the cover slip a few times until the root tips are uniformly squashed in between the slide and the cover slip. A properly squashed slide will appear faint cloudy pink to almost colorless. The root tip slide is now ready for microscopic observation. This is how a finely squashed root tip appears under 10x of the microscope. As you can see, the cells of the root tips are nicely spread out with almost no overlappings. If root tips are not squashed enough, you may repeat the squashing process until you get well spread root cells. Scan the slide for cells showing mitotic division stages under low power and then proceed to observation under high power, such as 40x or 100x oil immersion lens. For observations under 100x objective, you will need to add a drop of immersion oil on top of the cover slip and gently lower the objective lens until it touches the oil surface. From there, use the fine adjustment knob for focusing. You may take micro photographs of the various stages by employing a simple technique using a mobile phone and a tripod like so. And here's a quick slideshow of the various mitotic stages of cell division in root tip cells of Allium species.
Okay, that's it. Our presentation for today's session. The next session will be Q&A. If you have any question, just unmute your microphone or type it in the chat box. Thank you guys for your attention.